In the mid 1970s, and 80s, many powerful new methodological biotechniques were developed, which allowed us to carry on certain functions to reach the above aspects to the maximum possible extent in a positive approach. And these techniques provided the foundation of new biotic or molecular biotic. So now we are learning about seventh unit. So it belongs to biotechnology. All of you try to learn, students. So now we are discussing about concepts. What are the concepts? Okay, especially these biotechnology techniques. Okay, it is provided the foundation of new biotic or molecular biotic. These are recombinant DNA technology and cell fusion and bioprocess and structure-based molecular design. These are all those things very important. And physical traits are coded by basis of DNA, which are found in human chromosomes. In the 1970s, man became successful in a new techniques which allowed to remove genes from one organisms and insert into another organisms. So a real world example involves taking the human interferon genes and putting them into the bacterial E. coli. Interferons were too scarce to be used as drugs till the invention of biotechnology of interesting genes. However, E. coli is capable of making mass quantities of interferons. In the present day, interferons are most important drugs to treat cancers like hairy cell leukemia and corpus sarcomas, and also used for viral infections like hepatitis C and B. The cell fusion biotech uh, technique has led to the development of a monoclonal antibodies like mouse cancer cells, like this is also called as myelomas. Mouse cancer cells called as myelomas that grow indefinitely can be made to fuse to mouse WBCs. WBC is nothing but white blood cells, immune lymphocytes, and which can produce antibodies. So whenever myeloma cells can be, these are cancer cells. Cancer cells are the most important character of cancer cells is indefinitely they can produce, they can replicate. The cell division is continuously occur. So whenever we fuse the immune lymphocytes to these myeloma cells, which produce antibodies, the result is a hybrid myeloma cells called hybridoma, which has characteristic of both parent cells and cancer cell character at the same time WBC characters. So and most important is that these structures are immortal, behave like a cancer cells and produce antibodies like WBCs. So both parental characters are there. The cancer cell character like it is immortal, it is continuously divide and redivide and it forms many cells, number of cells. This is one of the character. It is the character of cancer parent cells, and one more parent is WBC cells. They can produce the antibodies. Whenever we fuse it to these two cells, so this shows two parental characters, like it behaves as immortal. At the same time, it produces continuously antibodies like WBCs. So hybridomas are important because they make monoclonal antibodies. So monoclonal antibodies, they are produced by this technology. Monoclonal antibodies have revolutionized medical diagnostic testing, and most useful technique has come out from this. Biotechnology can be performed by any Lebanese pregnancy test. So these are all those things. These monoclonal antibodies involved in pregnancy test. Okay. A group of techniques called bioprocess technology is series of engineering technologies which allow genetically engineered cells and hybridomas and other types of cells to be grown in large enough quantities for the production of drugs for the uses of mankind. So biotechnology has engineered mammalian cells produce TPA. TPA nothing but tissue plasminogen activator or PLATA. It's whatever it is, plasminogen activating tissue. So this biotechnology has engineered mammalian cells producing TPA, tissue plasminogen activator. It is a protein involved in the breakdown of blood clots which is serine protease, and became important anti-clotting drug used to treat heart attack and most recently strokes. So it is one of the important applications. Nowadays, we know that strokes and heart attack is occur due to the blood clotting occur in the heart vessels. Otherwise, those blood vessels carry the blood to the heart. Whenever some clotting occur, so due to the clotting, what happened? It interrupt the blood flow. Blood is not moved and blood is not reached to the heart. Thus, it leads to the heart attack, otherwise stroke. So these to prevent this heart attack and resist the strokes, anti-clotting drug is there. It is yeah, it is a actually serine protease. It acts as a serine protease. It is an anti-clotting drug. It involved in the breakdown of blood clots. So next, structure-based molecular designs and computer-aided drugs has evolved in the late 1980s and is considered a biotechnique. So the structure-based molecular design technology paved a technique where an enzyme or receptor is the lock, the substrate or drug is the key based on which shape is designed with the help of computer. This has led a method in design novel new drugs. A disease can be treated using drugs which can block enzymes or receptors which are placed at key positions to cause disease. In example, the active site of the HIV protease, an important enzyme for the growth of the AIDS virus. The drug stops the viral protease from its functioning and is one of a major new class of anti-AIDS drugs called protease inhibitors. HIV protease inhibitors are peptide-like chemicals that competitively inhibit the action of the virus aspartate protease. These drugs prevent proteolytic cleavage of HIV, GAG, and polyproteins that includes essential structural and enzymatic components of the virus. And scope. Biotechnology is the science of the controlled application of biological agents for beneficial use. So science, biotechnology is not an independent discipline. Its well-known integration with allied fields such as biochemistry, molecular biology, and microbiology facilities the technological applications of biological agents. So medicinal biotechnology. Red biotechnology deals with production of medicinal drugs, which can be proteins or nucleic acids like DNA or RNA. There is no single environment of chemicals in synthesis as they are derived from microorganisms which synthesize naturally. The first approved product for therapeutic use was biosynthetic human insulin made via rDNA technology. Here, human insulin replaced the pig insulin that was used previously and revolutionized the industry with its success. The human insulins called RHI, or the trade name Humulin, was developed by genetic but licensed to Eli Lilly and Company, which manufactured and marketed the product of Certin in 1982. So it is one of the important, red biotechnology and whatever it is, deals the drugs, production of drugs with DNA or RNA. So there is no single involvement of chemicals. What is that? Chemicals are not involved. Synthetic or whatever it is, the chemicals are not involved in the synthesis of this type of drug, and it may be derived from the microorganisms which synthesize natural. It's natural synthesis occur. It may be sometimes they are involving the microorganisms. The first approved product is we know that the first biosynthetic drug is human insulin. It is made via rDNA technology. So that's why rDNA technology is very, very important. So through the rDNA technology, first human insulin is synthesized to cure the diabetic peoples. So this production first licensed to the Eli Lilly company in 1982. So this human insulin, it is synthesized human insulin called as Humulin. It is also called as RH1. The trade name is Humulin. You remember that. So how it is synthesized? Already we discussed that the human insulin is replaced the pig insulin that was used previously and revolutionized the industry with its success. So another technique is the gene therapy, which deals with diagnosis and treatment of genetic diseases and some other diseases such as cancer. This therapy encompasses the manipulation of genes and the correction of defective genes. During this process, genes are inserted, deleted, or modified. So one of the most common forms of gene therapy is the incorporation of functional genes into an unspecified genomic locations in order to replace a mutated and dysfunctional gene. So industrial biotechnology and biotechnology and environment, these are all the applications. The first one is industrial biotechnology. Industrial biotechnology was for the large scale production of alcohol and antibiotics by microorganisms. So we know that alcohol like beer and wine and some antibiotics like drugs, okay, we can produce from the microorganisms. So at present, various pharmaceutical drugs and chemicals such as lactic acids, glycerin, etc., are being produced by genetic engineering for better quality and quantity. So lactic acid and glycerin, all those things we can produce from the microorganisms uh, by genetic engineering. So due to this genetic engineering, we can get the better and quality and quantity. Biotechnology has provided us with a very efficient and economical technique for the production of a variety of biochemicals, example, immobilized enzymes. Next one is biotechnology and the environment. Environmental problems such as pollution control, the depletion of natural resources for non-renewable energy, and conservation of biodiversity, etc., are also being dealt using biotechnology. Bacteria are being utilized for the detoxification of industrial effluents to combat oil spills, for treatment of sewage, and for biogas production. So biopesticides is another safe method alternate to chemical insecticides, pesticides, which are being used for control of pest disease weeds. That's why in this we can whenever toxic effluents or industrial releasing some substance or effluents are there, these industrial effluents are detoxified by this technology, biotechnology. We can use some microorganisms and sometimes some combat oil spills are there. For example, the oil it is transported in uh, what is that in seas? We know that, okay, from one country to another country. So in seas, is there any damage after this oil it is form a layer in water, sea water, and it is harmful to the aquatic organisms. This is called as oil spills. This combat oil spills, it is detoxified, removed by this technology. And for the treatment of sewage of biogas production also, we can use this biotechnology. And biopesticides also is there. So biopesticides, it is another safe method alternative to chemical insecticides. So whenever we use chemical insecticide, it is dangerous to our health. It is harmful to our health. Okay. So instead of that, if we use biopesticides, it is safe and we can control pest, disease, weeds, and easily. And it is environmental friendly also. Biopesticides are environmental friendly. And biotechnology and agriculture. Nowadays, plant tissue culture is widely utilized for the rapid and economic clonal multiplication of fruit and forest trees, for the production of virus-free genetic stock and planting materials, as well as in the certain of novel genetic variations through somoclonal variations. With the aid of RNA technology, it has now become possible to produce transgenic plants with desirable genes such as herbicides resistance, disease resistance, increased shelf life, etc. Techniques such as molecular breeding have been employed to accelerate the process of crop improvements. For instance, molecular markers such as restriction fragment length, polymorphism, and simple sequence repeats provide potential tools for the indirect selections of both qualitative and quantitative traits and also for studying genotypic diversity. So next branches of biotechnology. The first one is red biotechnology, green biotechnology, and blue biotechnology. Red biotechnology, it includes medical procedures such as utilizing organisms for the production of novel drugs or employing stem cells to replace or regenerate injured tissues, possibly regenerate whole organs. It would simply be called as medicinal biotechnology. It's a red biotechnology, nothing but medicinal biotechnology. We can use that production of novel drugs. We know that nowadays we are producing new drugs with the help of biotechnology, and we can employ the stem cells. Stem cells are self-renewing cells. Okay, these stem cells it is replaced or regenerate at the region of injured tissue, and these all those things. And due to this, the total organ is what is it regenerate. So this is used in this technology. This is called as red biotechnology. And second one is green biotechnology. It applies to agriculture and involves such processes as the development of pest-resistant grains and the accelerated evolution of disease-resistant animals. Green, nothing but it is related to plants or agriculture, whatever it is. Due to this, with the help of this green biotechnology, we can develop the pest-resistant grains and trees, whatever it is. At the same time, disease-resistant animals also. And blue technology, so it is as name indicated, it is purely belongs to the aquatic organisms. This rarely mentioned encompasses process in the marine and aquatic environment, such as controlling the proliferation of noxious waterborne organisms. And next, white technology. So white, also called as gray technology. And this white technology involves industrial process such as the production of new chemicals or development of new fuels for vehicles. So nowadays, biofuel also developed. So it is due to this white biotechnology. At the same time, pollution-less fuel. So all those things, okay, developed by this white biotechnology. And non-gene biotechnology. It works with whole cells, tissues, or even individual organisms. So non-gene biotechnology is the more popular practice involving plant tissue culture, hybrid seed production, and microbial fermentation, production of hybridoma antibodies, and immunochemistry. So next one is gene biotechnology. It deals with genes, the transfer of genes from one organism to another, and genetic engineering. So next, unit 8 is molecular techniques in gene manipulations. It is also very, very important for theory exams. After studying this unit, you will be able to learn the importance of gene manipulation, and you can understand about cloning vectors and their importance, and you know about methods of cloning. So it is a method which manipulate with the genetic materials, which include gene splicing with the use of recombinant DNA. It results to form monoclonal antibodies or PCR, polymerase chain reactions. So for the first time, gene manipulation was used in the agriculture for the improvement of the quality of the plants. It was discovered in 2001 in Mexican corn, native Mexican corn. So the use of human genetics is still very complicated. Nowadays, bacteria is used to produce some human hormones. The gene that code the hormone is isolated and it is inserted into the genome of the bacteria, like E. coli. So then they are cultivated on the suitable cultures. The bacteria becomes capable of hormone production like insulin, human interferons, human growth hormones, and others. In another method of recombinant DNA, it allows us to produce the antibodies. The gene manipulation allowed us to create the gene maps of various organisms. It is carried in three steps, like to isolate DNA from the organisms, to insert DNA into DNA vector, and third one to transfer the vector by transfection or transformation into the host. Cutting up DNA molecules into fragments is with specific restriction endonuclease enzymes. These fragments can be readily joined together by activities of DNA ligase enzymes. Actually, the purpose of vectors was to propagate cloned DNA, but nowadays they are fulfilling other roles like facilitating DNA sequencing, promoting expression of cloning genes, and facilitating purification of cloned genes, its product, and reporting the activity and localization of proteins. And vectors are two types: cloning and expression of vectors. So cloning vectors are those which autonomously replicating genetic elements. These are used to carry a cDNA or fragment of genomic DNA into the host cell for the purpose of gene cloning. The DNA fragments formed by restriction enzyme digestion as such cannot enter directly into bacteria cells, but when joined to a vector, its entry into host cells is very easy. So the cloning vectors, now we have to learn the features of cloning vectors. It should be able to independently replicate it and carry DNA segments. It's replicated, self-replicated DNA segments called as vectors. Otherwise, it is plasmid. Okay, so whatever it should be able to independently replicate and can carry the DNA segment, and it should contain several restriction enzymes, cleavage sites, which are presently only ones in the vector. The same restriction enzyme is used to cleave both vector as well as DNA fragments to be inserted into the host. The next one is it should carry suitable marker to identify host cells that carry the vectors from the host cells that do not carry the vectors. And it should be easy to obtain vectors from their host cells. And depending on their nature and sources, vectors we can classify into following types like plasmids, bacteriophages, cosmids, and phasmids and artificial chromosomes. So plasmid vectors. These are naturally occurring extrachromosomal and double-stranded DNA molecules that replicate autonomously within bacterial cells. The replication of vector depends on the same enzyme which replicates the chromosome of host, and these are inherited to daughter cells along with host chromosome in cell divisions. The size of the vectors varies from a few thousand base pairs to more than 100 kilo base pairs. And plasmid vectors. Students, these types of vectors are very, very important. Definitely, you can get one long or short questions regarding these vectors. You all have to try to learn and try to understand carefully. The plasmid vector can be single copy, which is present as one plasmid DNA per cell. Otherwise, multi-copy plasmids also is there, multi-plasmid, which are maintained as 10 to 12 genomes per host cells. And third one is plasmids under relaxed replication control, which possess capacity to increase their number to an extent of 100. So for acting as cloning vector, it should possess the following features. These are, it can be easily isolated from their host cells, that is the first one, and it should have single restriction site for one or more restriction enzymes. We know that already we discussed, and insertion of a linear foreign DNA at one end of these sites should not alter its replication properties. And next one is it can be reintroduced into the host bacterial cells, and cells carrying the plasmids with or without insert can identified or selected. And Bolivar and Rodriguez, who discovered it. PBR is a vector. This PBR vector group of plasmids are the most widely used cloning vectors. Among these, PBR 322 is modified with different sequencing compared to earlier plasmids, and PBR 318 and PBR 320. And PBR 322 is widely used as a model system for the study of prokaryotic transcription and translations. So transcription and translation is the translation is a protein synthesis. Transcription is the formation of mRNA from DNA called as transcription. So this plasmid is 4,362 base pairs in length and has origin of replication that is derived from plasmid related to natural occurring plasmid like CO1E1. It possesses genes conferring resistance to antibiotics, example like ampicillin. And bacteriophages. These infect bacterial cells and are used as cloning vectors. Its small size, plasmids are best used for single gene insertion, and insert rarely be larger than 2 kilobases. These are not suitable as larger inserts will increase the plasmid size, making them unsuitable for transformations. So lambda phase as a vector is extensively studied bacterial virus as for DNA cloning. This vector has a head region containing the viral DNA genome and tail which functions in the infection, its host and E. coli. The silk type lambda virus particles has 50 kilobase linear double-stranded DNA having protein code. When virus attaches to the host bacteria, the protein code is shed and lambda DNA is injected into the cells. And it includes in bacteriophage includes two cycles, lytic and one more cycle is lysogenic cycle. There the lytic cycle is the viral DNA replicates initially bidirectionally and subsequently resulting in a rolling circle model in a linear multimers of the versions. The lambda multimers are snipped at cos sites to produce unit lengths of lambda genomes which are wrapped within the synthesized protein code. So some lambda gene products cause lysis of the host cell wall, allowing the virions to escape and infect new bacteria. In lysogenic cycle, the lambda genome possesses a gene at which has a homologue in the E. coli chromosomes. So opposition of the two antigens, what is that? It can cause the recombination between the lambda and E. coli genomes and subsequent integration of the lambda DNA with the E. coli chromosomes. In this condition, the phage DNA is called provirus and the host cell as lysozyme. So the phage DNA can remain stably integrated for long periods, but it has the capacity for accession from the host chromosome and entry into the lytic cycle. The genes controlling the lysogenic cycle or the phase are located in a central segment of the lambda gene. So, however, the decision of the phase to enter either lytic or lysogenic cycle is controlled by two regulatory genes. These are CL and CRO gene, which are mutually antagonist. So, in the lytic state, the CRO gene dominates and causing the repression of C1 and replacement of lambda vectors. The lambda genome contains some genes at the central segments that are required for the lysogenic cycle, but not for lytic functions. So therefore, replacement of lambda vectors can be prepared by replacing this segment with a DNA segment of interest. So using this strategy, foreign DNA up to 23 kilobases in length can be cloned in such vectors. Such vectors are normally used for making genomic DNA libraries. Examples are EMBL3 and EMBL4. These two vectors are designed in such a way a central non-essential part of about 44 kilobases long can be positively replaced by a foreign DNA. Cleavage of the phase with appropriate enzymes generates three fragments, left arm, right arm, and central fragment called stiffer. This central part can be replaced by foreign DNA of 22-23 kilobase per long. And insertion lambda vectors. So these vectors are used for making cDNA libraries, and insertion vectors are prepared by modification of the lambda genome to permit insertional cloning into the C1 gene. Example, lambda GD10 is a 43 kilo base double-stranded DNA for cloning fragments that are only 7 kilobases long. The insertion of foreign DNA inactivates C1 gene and generates C1 phase. So non-recombinants, this lambda, it is CL1 plus form the clonally plaquenil appropriate E. coli host cells. While recombinant lambda phase is Cl minus and form clear plaquenil E. coli host. So like that. And next one is COSMID vectors. It is also very, very important. And these are formed by inserting the cos sequence of a phase into a small plasmid vector. So the cos sites enable DNA to get wrapped in lambda particle and permit their selections as well as purifications. These cosmids perpetuate in bacteria and do not carry the genes for lytic development. The most important advantage of usage of cosmids for cloning is that is high efficiency to reduce a complete genomic library of 10 to the power of 6 to 10 to the power of 7 clones from a mere 1 micrograms of insert DNA. Its disadvantage is inability to accept more than 20 to 50 kilobase pairs of DNA. For example, PRG 930 centimeters. And phage mates vectors, it is also very, very important. It is formed artificially combined properties of phages with plasmids. It has a multiple cloning sites and include inducible lag promoters and origin of replications. And PB blue script IIKS is derived from the plasmid PUC190. These are names of the plasmids and is 2961 polylinker transcriptional of lag gene proceed from restriction site for KPNL to SACL. So this artificial vector has multiple cloning sites flanked by T3 and T7 promoters and an inducible lag promoters, lag1, and upstream of lag Z regions which complements with E. coli. It helps in selection of chimeric vector using the criterion of white clonies formed against blue clonies are formed if no foreign DNA is inserted. It has FL plus and FL minus origin of replication derived from the filamentous phase for recovery of sense like plus and minus antigen strands of lag gene where the host is conflicted with helper phase. The artificial chromosomes, bacterial artificial chromosomes, BAC vectors. So vectors used for cloning DNA in bacterial cells have high to moderate number of replicates. So these vectors yield very high number of DNA clones depending on replication vigor. Its advantages is structural stability of the inserts. This results in deletion or rearrangement of part of cloned DNA. It is common in DNA inserts of eukaryotic life. Thus, larger DNA in bacteria is very difficult to clone and maintain. So then vectors with low copy number of replicants were developed to overcome the site limitations. In a factor called fertility plasmid or F factor from E. coli is prepared. It has two genes for A and for B, which maintain the copy number of F factor at 1 to 2 per E. coli, which can accept large foreign DNA fragments. And the recombinant vector can be transferred into bacterial cells using electroporations. When such vectors are used, a low yield of recombinant DNA will be recovered from the host cells. So bacteriophage P1 and P2 derived artificial chromosomes vectors. Some bacteriophages have relatively large genomes and thereby can accommodate larger DNA fragments. One such is bacteriophage PI, which can package 110 to 115 kilobase pairs of DNA in the PI portion code. The P1 cloning vectors are prepared from components of P1 phase that are included in a circular plasmid. The plasmid vectors is cleaved to generate two vectors arms, two vector arms to which help which up to 100 kilo base pairs of foreign DNA is ligated and packaged into the P1 PI protein code in vitro. And artificial chromosome, one more is that it's very, very important. Yeast artificial chromosomes, YAC. Okay, so it's sometimes pronounced like yeah, otherwise yeast artificial chromosomes. So the construction of yeast artificial chromosomes provides more advantages over cloning in bacterial cells. It can clone very large DNA fragments. So artificial yeast artificial chromosomes has the following features. So these are it is telomere at each end. Yeast telomere is present TN at each end, and yeast centromere sequence CN, allowing the regulating the segregation during the mitosis, and a selectable marker on each arm for detecting the YAC in the yeast, for example, PLE and TRP1 and URA3 like that. Okay, for tryptophan and uracil independence in TRP1 and URA3 mutant strains is present. An origin of replications, ARS, autonomously replicating sequence, allows the vector to replicate in east. And distinction sites also, it is unique to the YAC that can be used for the insertion of foreign DNA. And cloning methods. It is also very, very important. Naturally occurring reproduction is always found in organisms since their origin. So since their origin, and which makes them to increment their progeny. It is the reproduction method used by plants, fungi, and bacteria, and making clonal colonies reproduce among themselves. Cell cloning. So, cloning of single cell organisms, first we have to learn, and stem cell cloning, those things are there. So, in cell cloning, it is nothing but production of monocells from single cell. For example, single cell organisms like bacteria and yeast adopt a very simple process of inoculation to multiply in number. But in cell structure cultures, cell will not grow readily a standard medium. A tissue culture technique for clone, distinct lineages of cell lines is involved in the usage of cloning rings. Here, a single cell suspension of cells is exposed to mutagenic agent or drugs. This agent drives for selection is kept at the high dilution to create a isolation. Stem cloning uses process of somatic cell nuclear transfer. In the stem cell research is called research cloning or therapeutic cloning. It is for just for harvesting stem cells, which can be used to study human development and to treat diseases. And animal cloning is there, so gene cloning is there, and this animal cloning, it is organisms cloning as a process of creating new multicellular organisms which are genetically identical to one another. This is nothing but a form of asexual method of reproduction, which is commonly practiced in horticulture since many years from in plants. Okay, it's another thing is dolly and sheep. It is the first mammal which is successfully cloned from an adult somatic cells, not from germ cells, not from sex cells. It is from somatic cells. So for this, a single cell from udder, it's nothing but it's mammary gland of female cattle, sheep, goats, horse, whatever it is. It's called as udder. So having two or more tests and having hanging between the hindlings of the animals. So this are 6 years old biological mother. So a dolly embryo was created by taking the cells and inserting into the sheep ovum. It took 434 attempts before a successful embryo. So that much of attempts are there. So anyhow, finally they get that after long time. So this embryo was kept inside a female sheep for normal pregnancy. This experiment was carried at Roslin Institute in Scotland by British scientists Sir Ian Wilmot and Keith Campbell. So Campbell. The biological mother gave birth to Dolly in 1996, which died in 2003 after 6 years. So Dolly suffered remains were placed at Edinburgh's Royal Museum, a part of the Nation Museum of Scotland. The first mammalian cloning has success rate of 29 embryos per 277 fertilized eggs, which produced 3 lambs at birth, one of which lived in woven experiment involving 70 cloned calves, and 1/3 of cows died at a young stage. The first cloned horse, and Prometea, took 814 attempts. Next, gene cloning also very, very important. It is nothing but molecular cloning, which refers to the process of forming multiple molecules. At this juncture, generally, cloning is used to amplify DNA fragments containing complete genes. Further, it can also be used to amplify and any DNA sequence like promoters, non-coding sequence, and randomly fragmented DNA. It is used in a wide array of biological experiments and practical applications ranging from genetic fingerprinting to a large scale of protein productions. In the process of cloning of any given DNA fragment, surely it involves the following steps. These are fragmentation, ligation, transfection, and screening or selection. The first one is fragmentations, breaking apart a strand of DNA. And second one, ligation, gluing together the fragments of DNA in a desired sequence. And third one is transfection, inserting the newly found pieces of DNA into cells. The third one, screening and selection. Screening or selection, selecting the cells that are successfully transferred with the new DNA. So seventh unit and eighth unit is completed. Students, next unit is unit 9, animal cell culture. We can learn in next class. Okay, thank you. So students, if you have any doubts, you can note down and you can ask in next class.